kick that off in time. But
Maybe I can. Okay.
oh, sorry. What? Go ahead.
I don't have slides here prepared, but maybe I could, at least to start with, I could bring up something like a web page or something.
Yeah. If you can share
Sounds
it,
good.
we're gonna help folks as well.
Sure.
We've also got Rainer here who's
That's cool.
joining us from London. Okay. And now we have Poreg as well joining us.
Hello.
Hi.
Hi there.
How are you?
Good, good, good.
So you're in London?
I'm in London now. Yeah.
Okay. Wonderful. Okay. So just to give you sort of a high-level overview about the Neurokernel, but I would have to bring up a page here.
That's okay. So you can share your screen if you put your mouse on the Hangout over on the left-hand side. There's a green screen with an arrow on it. That's share.
Yeah, the screen share, right?
Yeah.
You could also give us a link to the page if that's easier.
Right.
Whatever you like.
Yeah. Gee, let's see. Let's just go to the Bionet website. Let's see. So if I how do I bring up actually a page here on this?
I'd say bring it up in your own computer and then go to the Hangout and hit the screen share button, and then it should give you the choice to pick that browser window.
But I have multiple browsers open up. I mean, multiple windows open. Do I have to specify it in some way or what?
Yeah, once you hit the screen share button, it'll prompt you to pick one.
Oh, I see. Okay.
Should show up on the list.
All right. Sorry about this.
That's cool.
Okay. I'll bring it up in a sec. That's maybe the easiest way to go at it. All right. So I am now, I have this up here. I go back, I guess, to the Hangout. And select window, but it doesn't show me any of the windows I just opened. It shows me something else.
The Hangout window might need to be on the other desktop. Otherwise, if you just send us the link, I think I could like share it on my side, and then we can do it that way.
Okay, all right. So the web page is basically the maybe I should send it out. It's www.bionet.e.columbia.edu.
Okay.
Search. Let's say. Could copy this and share it.
If you can copy and paste into the chat, there's a chat window in the Hangout, and that
copy and paste it on what?
Into
on the screen share.
Yeah, there we go. Yeah, in the chat, there's oh, you got it already? Okay.
Yeah.
So there's right above the green box for screen sharing.
Yeah. I see this.
Chat.
Just drop it there.
Yeah, if you can put it in the chat. There we go. Yep. Okay, perfect. And now I'll share that screen on my end.
Okay. Sorry.
No worries. There we go. Okay.
All right. This is what it is. So please just slide down. Towards the end. This is the architecture there.
Oh wait. Oh, I see. I had two. I needed to show the other one. Sorry. Technical issue on my end. Okay. Architecture. Got it. Did everybody see that?
It's not what I see, actually.
Oh, no, it isn't.
Yeah, I don't see the architecture either. Should be red, green, and yellow.
Okay, that's it.
That's it.
Okay, so this is the architecture. The architecture is based on these 3 planes. So what is the basic idea? The basic idea is that we look at the brain of the fruit fly. The focus is only on the brain, okay? And we look at the brain of the fruit fly as consisting of some 40 regions called neuropils. And for instance, examples of this are the medulla and vision, lamina and vision. They're all neuropils. And the way one looks at this is that neuropils have input axons and output neurons. And in the neuropil itself, there are something called local neurons. And the way to think about this neuropils, in IT languages, that these are like chips. And chips have an interface. And through the interface, they communicate to the outside world. So the neuropils are essentially interpreted as being chips, and they are called LPUs, local processing units. And that's what you see in the application plane. So in other words, logically speaking, the LPUs are interconnected through what is called tracts, essentially through axons. And they have local processing capability, like the logic on the chip, which doesn't know anything about the outside world. And then there's an interface which interacts with the outside world. And so the basic thinking oftentimes is that one has a neural circuit, and let's say a neural circuit models the entire brain. Well, we look at it as being decomposed into units, somewhat similar to what you have, let's say, on a computer board. And those units are these LPUs. So if I step back for a second, the standard thinking is that you have a very large neural circuit which is being emulated. But we think that that way of thinking is somewhat limited, in the sense it's very difficult to scale it up. Number one. Number two, people participating in various biological labs, they tend to focus on specific neuropils. So there's people who are looking only at the retina, or so there are people only looking at, let's say, a piece of the olfactory system, maybe the olfactory sensory neurons or the antennal lobe. And so they can model these species. In principle, they could also possibly implement parts of it, but they don't know anything about the rest of the system. And so the thought behind this modeling we have is to say, well, look, in the end, it would be great to have them participate. They will also have a lot of data, and they could participate in terms of, or contribute in terms of data, but also in terms of implementation, or in terms of requirements. And so now, if we go back to the architecture, you see the top layer has the LPUs. And so this is what a programmer essentially sees as the modeling of the brain. And the LPUs are then mapped into the hardware, which consists of CPUs and GPUs. The thinking here is that the CPU level essentially deals with compiling and the control of the whole system. And on the lowest level are the GPUs, and the GPUs communicate directly. This is not how they were designed for. But this is where the technology is evolving. So if you would compare this with the architecture of the, in sort of very rough terms, with the architecture of the internet, the GPUs look like routers, and the CPUs look like the control of the routers. And then the top layer is the application layer, okay? And so this presupposes that the GPUs can do direct memory transfer. And the direction of technology is clearly emphasizing this, and we believe that we're going to see systems more and more directly interconnected. So right now, the link between the CPUs and GPUs is fairly strong, but I think what's going to happen is that the link between the GPUs directly is going to be strengthened, so that the CPUs appear essentially as controllers, and the GPUs appear as the computation engine. So this is the thinking behind all this. Okay, so on a conceptual level,
okay.
Let
me just break in here real quick, just for because I noticed we've got about 7 other people watching as well, and who just joined us. So for folks that are just joining us, we're talking with Dr. Aurel A Lazar, who's at Columbia, who's working on a project called Neurokernel, to do massively parallel simulations of neurons in the Drosophila brain. We are the OpenWorm project. Obviously, we also have a great interest in joining forces. We're also both very open source driven, and he's been walking us here through the architecture of the way that system works, so that we can understand the division between where graphical processing units, your video card, and CPUs, which are sort of mainly running your computer, relate to the sort of, what is it, local processing units is the LPU?
Yes.
The architecture of all these things are fitting together. And if you're watching and you have questions for anybody here, please on Twitter or on the Google Plus page for the project, and we would love to get the questions answered as we're going. So, okay, sorry, just so you were about to talk about the conceptual side.
Yeah, so conceptually, basically the architecture is divided into 3 pieces, and it's like a separation of timescales. And that's probably the most important aspect of this. On the GPU level, everything operates on the fastest timescale. And then there's the CPU level, and finally, the application layer. And so what happens is essentially that on the application layer, we have these LPUs, they're interconnected, and so what the programmer sees is a model of the brain. And the LPUs can be designed, and this is, we feel, a critical aspect of the project. The LPUs can be designed independently, provided that they expose open interfaces, which designers of the LPUs have to abide by. And so if you have these open interfaces, essentially one can then combine the LPUs in any way one wants to. Actually, the objects are really LPUs, and the corresponding sort of interconnects. What you see there, in a black box with sort of white little squares, those are patterns, or if you like, it's sort of in technology language, these are sort of switch fabrics. So it allows you to essentially interconnect the LPUs in arbitrary fashion. So now this is the programming level on the architectural level, and then of course there's much more to it, because not only that logically these pieces have to be put together so that a program can be executed, but possibly this has to be done with appropriate resource allocation. For instance, maybe an LPU might run on multiple GPUs, and the question is then how those resources are allocated and all this, and the interfaces would have to essentially communicate that information to the neurokernel, which would take care of all this. That part of the story has not been implemented yet. Now, we think that because of this openness, okay, what we have done essentially, we are saying we have given up, if you like, the control of software design in some ways, because we would like to have other people involved. They can do their own thing. For instance, suppose somebody comes in and says, hey, I'm interested in implementing the Lobula, and there are some other implementations there, but I don't like them for whatever reason. and that means, for instance, that maybe there's a different implementation based on different assumptions, maybe based on different models of neurons, etc. People in neuroscience tend to disagree on these things. So instead of arguing about what is the appropriate model, we are arguing that everybody should be actually accommodated. So if somebody has a different way to go about it and has different models, so be it. As long as the local implementation has the standard APIs, it can be then used by others and to see how well it works, and it can be compared with other implementations. And so we think that this is a critical aspect of this. We would like to have a library of implementations of all the neural pills, and the more the better, and people can compare them. This is one. The second aspect to this is that, of course, everything runs on commodity hardware. So we are showing here in this architectural representation lots of GPUs, lots of CPUs and all this, but this depends on the lab which is running it. And anyways, commodity hardware, the GPUs are, as you know, inexpensive, and they have this tremendous power to do simulations. And so we think that actually independent labs could run this. Finally, we also think that platforms like this could be actually connected to live rigs, where flights actually they're being recorded from, and one could actually then evaluate or validate, I should say, the code. So validation is biological validation. We think that the biggest challenge is biological validation, not program verification in the computer science sense. Program verification, has its own problems, but especially when it comes to parallel programming. But we think, as you know, we can deal with that. But when it comes to biological validation, that's much harder. But we think that that's really the challenge. And we think that credibility in all these implementations comes from there. So the question is how to do this. And so it's not enough that people with IT background get into this. We have to involve actually the biological labs in this. And we hope that this is going to be on some time frame possible. It's very tricky. So if you have some questions, please go ahead. This is sort of a very brief, high-level view. Looks like we lost.
Have we lost Stephen?
No.
He's back.
This is
okay.
excellent. I think there's a bunch of jumping off points here.
Stephen, we're having a hard time hearing you here. I don't know if it makes sense to maybe type out what you're looking to say. Oh, we've lost him.
He's actually lost. Okay.
Well, while we wait for him to come back, I just wanted to point out that, my immediate observation in listening to the presentation, the high-level presentation, is just how many parallels there are with the OpenWorm project, because we have precisely the same concerns where we want the project to not be just populated by IT people, people with an IT background, with software development background, but also include people with a biological background, so that biologically relevant data is included in the model. Also, the idea of separating the various models from the modeling framework, so that various competing models could be tested. That's definitely something that we've certainly thought about with the architecture of OpenWorm as well. So yeah, I'm struck by the number of similarities of the two projects, for sure. Very interesting.
Great.
Porig, I don't know if you want to say something.
Could you just give an overview as well about the use of GPUs? So you're using CUDA?
Yeah, we're using CUDA, and actually we are using PyCUDA, and we are using Python. So the programming is in Python, PyCUDA /CUDA. And you know, we have seen that other people are using OpenCL. That's another possibility, obviously. What we noted is that the implementations of NVIDIA of OpenCL, you know, they are somewhat sluggish. And so, you know, but in principle, there's no showstopper, really, strictly speaking, on this. So I don't know how you guys feel about this, but right now that's what we're doing.
Yeah. Okay. I mean, I think one of the things, I'm not very familiar with these packages, but I know I get the impression that OpenCL, it's much easier to switch between GPUs and CPUs for the target platform, whereas if it's hard-coded for CUDA, it's going to be specifically on the GPUs that it will run. Is that correct?
Yeah, there is some additional capability in OpenCL. Basically because you can program both the GPU and the CPU. Yeah. And so there is additional capability, but then, you know, at the same time, the NVIDIA folks, they're pushing this very hard. And it seems to us that there are some features which CUDA provides, but they are not available yet in OpenCL. Yeah. So it's sort of a, you know, a situation where you know, both groups, if you like, you know, they're pushing this, and from our standpoint, you know, it shouldn't matter, okay, in the end, okay. Yeah. Here in some classes, you know, we're using OpenCL, and others we're using by CUDA, so you know, it's. But right now the implementation you see on the web is not using OpenCL. Okay. Yeah. But how would Neurokernel compare to some other platforms which use or neuronal simulators which target GPUs as a general purpose neuronal simulator for targeting GPUs? So, say for example you wanted to build a network, you wanted to simulate a small network, something like C. elegans, but you don't necessarily you want to wrap this in local processing units. So if you have basically just one local processing unit and don't use any of the infrastructure for connectivity between these, how would it compare to others in terms of building the connections and so on, just using a single LPU? Well, you know, we're sort of doing this both this. Okay, for instance, you can use an LPU, you can run it under the Neurokernel, or you can run it also separately. Okay. Yeah. For instance, if you want to test it and all this, you know, we are also sometimes just running it separately outside of the Neurokernel. Now, fundamentally, the difference is that we are essentially saying that the architecture is partitioned into these building blocks, which are the LPUs. Yeah. And essentially what that means, or at least in principle, you could run an LPU in Sydney and the other one in London, and the third one in, you know, in New York, and you could run a program across the world, so to speak. Although this is not how, you know, we thought it out, but I'm giving this as an example that in the end, it's a completely distributed program. Okay. Yeah. And now it has these costs because as soon as you start doing that sort of thing, there are delays in it, and you know, things get a little bit more complicated. But essentially, the difference is that we're not using a monolithic program. And so, if you like, the direct answer to your question is, where is sort of the fundamental difference? I think the fundamental difference is that we don't have a monolithic program. In order to avoid that, we had to then, you know, provide these APIs, these open interfaces, and that provides huge flexibility in terms of scaling. So scaling now, which goes in the direction not only that we, in principle, we can use this for the fruit fly, but we could go to the housefly, was, you know, actually, you know, the whatever, sort of essentially bigger systems. And the system, at least, you know, from a design standpoint, appears that it would be scalable. It's a little bit more complicated, obviously, you guys know this, but the idea is essentially to deal with scaling up front.
Yep.
So there's no uniform way to design everything. You see, what happens is in the typical implementations, people say, well, I'm using some kind of a neural model, and that typically is across the entire system. For instance, take integrated fire neurons, okay? So you have 1 million of those, okay? This allows you, essentially, if you have the 40 neural pills, to have whatever implementations you want to. At the minimum, you could have, 40, meaning, different 40 different neural models, each one in different neural pills. That we feel it's important because, in neuroscience we tend to disagree on models, okay? And so just build it in that you have the flexibility, and that's that.
Can I ask you something
Sure.
relating to that? So if you have so many models, how are you planning to discriminate between them and decide which one is more accurate than which is the most accurate, for example? With our project, with OpenWorm, we're able to connect the connectome that we're modeling to the worm itself and then observe the simulated worm's behavior, and then we can compare that behavior to recorded behavior of actual worms, and then statistically compare the two. And that's that's the piece of the project that I happen to be working on, called movement validation. So I wonder if in this simulation, are you just simulating the brain? So obviously this brain is not manifesting as a simulated fly flying around. So then how are you able to discriminate between the models?
All right. So this is a very good question, and there's no definite answer to this. Okay, as you know, it's very complicated.
Right.
But I would say this: right now, the belief we have is this: that going from neural circuits to behavior, this is a bridge too far for the fly. Okay, we are looking essentially at neural circuits and computation. So we are looking at this is an information processor, which we are trying to understand how it works in terms of computation abilities. So now the demos we started making is, for instance, in vision, is that you present a video, let's say, to the fly eyes of some kind, synthetic or maybe some scenes in the forest or whatever, so natural scenes.
Right.
And then you start looking at various neural pills and see how this is processed, and you can visually inspect it, or of course you can do some evaluations, some statistical evaluations about it. The point is, though, that we can actually observe how information is being processed as it goes through various neural circuits. So the evaluation, if you like, becomes fairly powerful because you start getting intuition about it. And it's not clear, what, let's say, the early visual system is exactly doing. You start looking at the neuron, you start looking at the output, you start getting some insight about it. Now, this is sort of a computation idea, and you evaluate essentially what is computing the circuit. Okay? But from there, let's say, to decision making, to behavior, there's just huge gap there.
Right.
And so right now, we're saying, I would say this, we're not as ambitious, okay, because the system is very complicated, and we're not actually sure exactly how to bridge that gap to behavior. And by the way, this is a view which is held at least by some people in neuroscience, that the gap there might be too big. So now, since we have sort of a IT level conversation, this is like you take a smartphone and you look at it, at the circuits, means you look at the chips, and then somebody says, but how about behavior? But that means you look at the applications. So now you have an application, which is a piece of code, which you would like to relate it to the chips. There's no way to do that. It's out of the question. It's just not in the cards, okay? At least for us, from an IT standpoint. And so we also believe that now with the fly, the situation is similar. The gap is just too huge. And so we think that the first step is, well, we have some chips, let's see what they're doing somehow on the quote-unquote low level. Now, computation, usually is not looked upon as being at the low level, okay? But in this case, behavior is, okay? And so, in your case, I understand it perfectly, because, basically, you focus on the entire organism, and you see movement, and that's behavior, and you would like to relate it to maybe, some phenomena going on in the brain itself, right? Now, in our case, we are focusing only on the brain at this point, not on the organism as a whole. But that's not to say it would not be interesting. It would be very interesting, but we think that that's sort of it's a long way for us.
Well, to be fair to you, you're dealing with 3 orders of magnitude more neurons to deal with, so it certainly makes sense that the behaviors are getting to that level is a lot harder. Maybe from a software perspective, it's the difference between white box testing and black box testing, where you're actually looking at the neurons themselves, and verifying that, as you say, like the individual components are receiving input and producing the right output, whereas we're sort of treating it all as a black box and trying to see if the behavior that's manifested is correct. So, interesting.
Right.
This is a place, I don't know if you guys can hear me again.
Yeah.
We can hear you.
But this is a place where I thought, there could be an interesting dialogue between our 2 projects in that, our approach to doing the behavioral validation of the worm model, something that's at this point more practical to do with a worm model than a fly model, but as Aurel is already thinking about, comparing this to behavior of real flies, there might be a way in which there's inspiration that can be taken from our approach, and shared. And so I don't know, Michael, if you just want to say, like, in 3 easy steps, slightly more than what you've said already about how we do that, and I wonder if there could be a metaphor that could be made here, to the fly.
Sure. Yeah. I mean, I think the key is that the output of our simulation is an actual physical worm that we can. So we have the skeleton of. It's moving around on a simulated agar plate, and then it's that information about frame by frame where the worm is moving that we're able to compare to a real worm. And then the way that works is you have a video of a real worm that's been taken, and you use computer vision to segment that video into a skeleton, a wireframe of the worm's position, morphology at each frame of the video, and then from that we calculate features like the velocity of the worm, the amplitude of its oscillations, and with those features calculated for both the simulated worm and for the real worm, we statistically compare them and try to see if we can reject the null hypothesis that they might be coming from the same worm itself, and so that's how we're able to statistically compare the 2. And again, it's all from the behavior of the worm rather than looking at the actual neural circuits itself. Now that's of course not mutually exclusive. We could also be doing other testing. I haven't dealt with that myself personally, but I think that we could also be doing a lot of the testing that Neurokernel is doing also by looking at the actual neural circuit itself and seeing if it's producing behavior that makes sense. Or sorry, producing activity that makes sense. Pardon me?
Sorry. In other words, you're considering connecting actually the brain itself to control its movements?
Yes, exactly. Yes.
And so you started working in that direction, or right now you're just considering it?
I would say yes. We've definitely we're developing in that direction. We have the system at the point where just in the last few weeks, I think Porig is a big part of this part, connecting the what we call C302, the model of the connectome itself with the actual physical engine that is modeling the muscles and the movement of the worm in a physical simulated environment.
Uh-huh. So in effect, you're looking at it as being a robot. Is that correct?
A robot in a virtual reality, if you will. Yes.
Yeah, of course, in a virtual. But you're thinking about a robot now. So we looked at this a little bit, okay? But you see, what we're emphasizing is to what extent we could possibly connect our system to a rig.
Okay.
And to what extent we could actually validate, processes in the brain of the simulator with processes in the real brain. And so that means the emphasis is towards sort of biological validation. And in some ways, these things are very similar, but there is a difference there in the sense that one is sort of more emphasizing the biological aspect, and the other one is emphasizing more the sort of biorobotic aspects, maybe. Okay, is that something you share?
I don't know if I don't know if Porg wants to jump in and comment on some of the other things that he deals with there, but I can certainly say that, as I say, it's not mutually exclusive. So what we're doing is certainly not what I'm doing with movement validation is certainly not the only kind of validation that we'd like to be able to do.
Yeah, I mean, we're definitely taking a kind of a staged approach to it. So we're building the infrastructure at the moment to allow us to simulate the body of the worm, simulate it in an environment that's been going on for a number of years now, and we have a reasonably good fluid mechanics simulator, which can...
Beautiful stuff.
Yeah. So, I mean, we're basically at the stage now that we're building from the bottom up the neural network, making lots of approximations to begin with, but incorporating a small amount of detail in the neurons, as much as we know about the connectivity as possible, and then trying to get a framework where we can overlay that on the 3D simulation and get some interaction between the neural network and the 3D simulator. What we want to do then is gradually increase the level of physiological detail in each of the neurons and kind of insert it into that framework whereby, okay, we can do lots of tests with the integrate and fire neurons. Maybe we have some point neurons with ion channels. Maybe we have some multi-compartmental neurons in a few months. But have this framework so that we can have placeholders for putting in that greater levels of physiological detail. And when somebody does come along and says, okay, I do have recordings from whatever calcium dyes in a particular neuron, we can tune our model to incorporate that level of physiological detail, but at the same time have that full circle of, okay, we know which parameters we've put into our model, we know how it will run when we connect it to the physical model, and try to iterate a much better approximation of the behavior of the worm, and comparing that to the experimental data will be the kind of end goal of what we actually want to try to do.
I see. So you have a stage plan to bring in the brain, basically.
Yeah. So I mean,
that's what I'm hearing, at least.
Yeah, I mean, this neural network as well can potentially be run independently of the run and analyzed independently of the 3D simulation. So, I mean, we're building it with NeuroML, and the whole idea there is that it can be run on its own. But obviously, if it's doing something like causing oscillations in the muscles, then it would be nice to look to see whether that will produce a realistic behavior. But potentially, if somebody's interested in some other sensory pathway, there's nothing stopping you just running the neural network by itself, or just making a smaller network with maybe 10 neurons and trying to get that as physiologically realistic as possible, with a view towards feeding it back into the large-scale network.
So right now you have this movement, and so you have a control system, but you don't have a sensing system.
No.
To give feedback to the control system to maybe change behavior. Is that right?
That's correct at the moment, but it's hopefully not too far away. So I mean, they are pretty independent code bases. This so cybernetic is what it's called, this 3D fluid mechanics simulation of the worm body. So it is quite straightforward to actually sense at various different points in that 3D body that, okay, the nose of the worm has been touched, for example, and the slightly difficult part will be to relate that back in real time to the particular neuron which would be interested in that touch sensation. But
yeah.
But
we would like to transfer all the tasks, essentially, what you have now right now, this movement to a controller, which would be the brain of the worm, and then at the same time you have to have also some sensing capability so that decision making can take place in the tiny brain, right?
Yeah. So I mean, we want to be as data driven as possible. So only actually put at the very minimum input into one particular neuron that's known experimentally to be the touch receptor, and then from there try to tune the network so that activation of that particular neuron will result in maybe reversing of the motion of the worm. So I mean, there are some very well studied pathways like this in the worm, and we'll be looking at some simple well studied examples of those to begin with. But I mean, in practical terms, one of the issues here we have is that it's a C++ code base for the cybernetic simulation. It's probably going to be Python or Java that's going to be the neural network will be running in. So I mean, in practical terms, it's maybe hooking up these independent code bases rather than making it as efficient as possible to send spikes back and forth between these or some other scenario. So I mean, there's just practical concerns about hooking up very different simulators.
Right. Aurel, can I ask you a question?
Sure.
So the right now you're focusing on modeling the visual system of Drosophila, as I understand. Pardon me?
And olfaction.
And olfactory. Okay. So that's basically what my question was going to be relating to the input into your system. So I guess in our case, C. elegans doesn't really have that much in the way of sensory input, at least compared to the massive amount of visual data that the fly might experience, as well as, I guess, olfactory. So maybe for that reason, it's been less of a priority to provide to complete that feedback loop and to provide sensory data into the connectome. For your project, I'm curious to hear more details. I know you spoke a little bit about maybe a forest setting, maybe some kind of visual data that you could also provide to a real fly. But how is that data being routed into your simulation, and what does that data look like?
Okay, so basically what we do is we generate videos of various kinds where what we do is we mimic some of the experiments. What people do is, with fruit flies, they do experiments with LCD screens, which are sort of curved. And so, the fly sits on top of a ball, possibly, is pivoted, from the top, and so he can sort of walk on the ball, but basically it has restricted movement. But it sees an LCD screen. And on the LCD screen, you can generate whatever you like, videos, or maybe some bars moving along, what have you. And now, the eye itself, on the retina, it's a compound eye. There are something like 768 ommatidia. So it's like small little sort of eyes, if you like, okay? And each of them has 8 neurons, or photoreceptors. 6 R1 to R6, that has to do with, essentially, if you like, sort of black and white, and then 2 for color. And each of them does transduction of, essentially, let's say, the light intensity into a current. This transduction is taking place sort of on the molecular level, basically, and we have differential equations to describe this. This has been studied in detail. Essentially, this transduction takes place in sort of small microvilli, and each of them basically is described roughly like 20, let's say 20 equations, and there are maybe 30,000 microvilli associated with each photoreceptor. So you start getting a pretty huge number of equations, and they typically drive then a point neuron model whose output, however, is graded potential. And this is where the headaches start, because the entire system is basically analog. So there's no spiking. There's no spiking, at the output of the retina. There's no spiking at the output of the lamina. There's no spiking at the output of the medulla. There are only, later on, there are some neurons which spike. And so everything is graded potential. And it's similar from that standpoint to what's going on in a worm.
Yeah.
And that is difficult actually to conceptually understand, because analog electronics is still an art, digital is what is here. And so you see what happens is one model is to implement all these equations, and then actually you can visualize exactly the output of the retina. You can see exactly how information in the visual space is transformed, which is essentially in the optical space, optical frequencies, is mapped into electrical currents, and that you can visualize. And you can look at it. You look at it, qualitatively, how good this is. And then you go into the lamina, and the lamina has the same retinotopy, meaning, there are circuits which roughly they're driven by an ommatidia. It's not exactly the way it is, but I think that was sort of, in the first approximation, that's the way to look at it. And so you have sort of a parallel programming model there. Ommatidia, there are cartridges in the lamina and there are columns in the medulla. And you start seeing the processing being done on this. You get a sense of what's happening, in terms of number of neurons. The cartridges have something like 10 neurons each, and there are also some interconnects. Then you go into the medulla, there may be up to 300 neurons on each of the columns, and then the interconnects between them. And the problem is that, so we have information about this, but it has a lot of gaps. So it's not like is the case with the worm, where, let's say, exactly how many synapses you have, exactly how many neurons you have, etc., etc. Here we have, and exactly the connectivity. Here there are a lot of gaps in this, and in particular the difficulty is that there is a large number of neuron models. I should say neural classes. I'm talking about now sort of the biological neurons, and that is also not clearly understood. Okay, so people know just by looking at the morphology that the neurons are different, but the characterization is not available. So there are a lot of gaps in this. From our standpoint, that means that one has to come up with some hypothesis and, try to understand what are the computational capabilities of circuits like this.
There certainly are data gaps on our end as well. I mean, although it's nice to have the full connectome, we still don't know the I/O characteristics, if you will, of each of the neurons. We don't know if they're inhibitory, if they're excitatory. So that there's certainly a major lack of data there too. So even if you had the connectome, it certainly wouldn't be sufficient to completely finish the model.
I do want to know though, how are you coping with some of those gaps? I think that's another thing that we share in common. So I mean,
Right, yeah.
Are you able to encode proportions of cell types into the circuits? How are you dealing with connectivity? Are you doing it on with probable? I would assume the first most reasonable thing is some sort of probabilistic connectivity.
No probabilistic anything.
No. Okay.
We are using connectivity. This data is available, and it's SEM data, and that's what we're implementing. Now, the gaps we have is that we know about synapses, but we don't have a characterization of the synapses on the neurophysiological level. And also, we have zero characterization of gap junctions. In fact, we don't know where the gap junctions are. That is not available. Okay, and gap junctions in vision are very important because most likely they are needed for very fast processing. There are no time delays going through gap junctions. There are time delays when you go to chemical synapses.
Right.
And so the models we are using lack gap junctions, but they have anything else. And we don't make assumptions about connectivity. In other words, it's not stochastic connectivity. But there are some gaps there. You know, sometimes you look at a neuron and you're not sure, is this an input neuron? Is it an output neuron? Is it a neuron which, you know, comes from when the medulla gets into the lamina, it's feedback neuron? You know, there are some interpretation questions there. But by and large, we look at this to be, if you like, the connectome as being fairly established in parts of the fly brain, like the lamina and medulla, at the level of synapses. In the rest of the brain, we have only mesoscale information, which means essentially that when it comes to the rough connectivity between various neuropils, that's well understood. But when it comes to exactly synapses and where they're located and all this, that is guessing. So there are two methodologies, in other words. There's one methodology which, you know, the group, let's say in Hsinchu in Taiwan is using, where there's a mesoscale connectivity which they worked out. So mesoscale means that you don't know exactly where the synapses are, but you sort of see where the neurons are coming together, and you start making guesses how many possibly synapses are in between them and all that. But actually, there's huge gaps there. Okay, and then there is this very detailed information using SEM, and there you know about all the chemical synapses, but you don't have information about the gap junctions. So there's still, you know, way to go there.
So if any budding graduate students or postdocs who are looking to have an impact, locating all the gap junctions in Drosophila brain would be a hugely impactful.
Yeah, but you know, this SEM data took like, you know, for the lamina and medulla took like 5 years, I think.
Yeah, it was like 13 from the C. elegans.
Yeah. So you know, yeah, we have to live with this. I wish we had better data, but you see, in a sense, this should not be a showstopper for us.
Okay.
You know, we have to push. Essentially, I think we have to push the field, in spite of the fact that there are many gaps there.
I agree. I would be remiss if I did not, at some point here, just mention the C. elegans project, just real quick, and I think I might have mentioned to you verbally when we last met, Aurel, but just because there is another project that does have relations to hardware, and on the C. elegans side, it's a EU-based project that is reproducing neuron models in FPGAs. It's a consortia that I guess was funded by the EU last year, and we're continuing to interact with them. And I guess it's probably something that will proceed in parallel, and you know, until it's further down the road. But some of these ideas of how do you abstract away the neuronal model from the hardware model that you're making here, I think there's an opportunity to cross-pollinate. I don't really know actually what their architectural strategy is for doing that, but I know that Finn Kruer from the C. elegans project has been talking with Porig here, intersecting on a LEMS way of describing a neuronal model, which is maybe maybe slightly more detailed than I don't know than the models that you're using, maybe, but it's a third project that comes to mind here in the same space that's pretty related to this. We're also looking to potentially have a joint workshop with them next June at the biological C. elegans meeting, where there's an international worm meeting basically of all the biologists, and that should be another interesting opportunity. So I don't know if you've talked with those guys yet. Are you aware of those guys?
I'm aware a bit. I don't know the details.
Okay.
I noted this, that there are efforts in using FPGAs, and my understanding is you know, they're used as accelerators the same way as we're using GPUs. And it would be actually, I think, interesting to have a common programming environment so that whether you run things on GPUs or you run on FPGA, it shouldn't matter.
Yeah.
I don't know, did you guys give it some thought about this?
So on our side, I think only so far as using OpenCL, as you guys were talking about before. But yeah, so the idea would be there that it's independent of those two. But that was, yeah, so I think it'd be interesting to build community outside of just OpenWorm to think about this as simulators in general that have that ability. Because I mean, the architecture that you're proposing goes a few steps beyond just OpenCL. I mean, it's at a different level of organization. So that could be an interesting next thing, actually.
Yeah, I mean, you know, in principle, you know, the type of hardware which runs at the low level, you know, quote unquote, it should not matter. Okay, it's easier said than done.
Right.
Okay? It's much more complicated than that. But you know, I think that is quite attractive actually, because obviously with FPGA you have, you know, some things you can do some things you cannot do with GPUs. In particular, you know, if you want to put things on a robot or something, where size matters, and you know, you can have some sort of very compact, you know, solutions. And when it comes to developing software, you could do it from the same environment, basically. I mean, at least, I mean, I don't know what the other folks here think about this, but you know, Patrick, what do you think about this?
I think just from the approach we're taking in OpenWorm, it's really concentrated on specifying the physiological structure of the network first, and then worrying about the implementation details later. So what we've got is some Python scripts to describe the structure in Neurokernel, the connectivity, and then developing other pipelines excuse me, for mapping this to simulators like Neuron. And this is exactly what Finn Kruer in the Science project is doing. He's taking the same network in Neurokernel and LEMS and trying to map that onto the FPGAs. So, I mean, it's completely conceivable that we could use the same approach for mapping this onto Neurokernel. So we have the access to the descriptions of the neuron models in LEMS, and I think some of the guys there have looked at it already to map, for example, the Morris-Lacar and integrated fire cells to Neurokernel. And it could conceivably, if there was a generic mapping from LEMS to Neurokernel, I just give a quick look through your code, and I think basically what you do there is generate the function calls, the CUDA specific code for each neuron model, and you could potentially generate code like that from the LEMS descriptions. And if it was generic, then you could generate the connectivity and so on take it out of the Neuromel files, put it into the an equivalent in Neurokernel, and then run it on a GPU much faster than we can on Neuron, say, for example.
Yeah, we're interested in definitely moving things in the direction of NeuroML. Okay, right now we are using an XML-like, you know, format, and we think that actually that should be not a problem. It's a question of time and all this, and I think that definitely in the long term, or whatever term, we ought to sort of move in the direction.
I wonder if there'd be somebody in, you know, either of our communities that would be interested in trying to, you know, import the C. elegans connectome into the neurokernel core, essentially, and just like, I imagine you have some way of defining the connectomes, as you've just said. There's implementing NeuroML, but then there's also just like seeing if we could run it and seeing what kind of output we could get. I think that would be an interesting crossover project.
Wow,
because yeah,
How big a project would this be? What do you think, Stephen?
Well, it's just a matter of translating the NeuroML specified connectome to the format, the XML format that you're currently using. Then that's perhaps the relatively straightforward step.
Yeah. Do you have documentation pages on modifying the?
I'm sorry. Can you repeat that?
Do you have like a documentation page that explains how you would modify any of the connections on your system? So I'm thinking like a set of instructions of like you have to download this, you have to go into this file, you have to change this thing. Because if we have basic instructions on how to change the connectivity patterns of the neurons in your system, then that would be the first thing you'd need to then pull in whatever way the C. elegans connectome.
Okay, so look, there's a slight difference there. What's happening in this, we have that on the architectural level, but we do not have that on the neuropil level, the LPU level.
Okay.
Because on the LPU level, everybody can implement whatever he wants.
Okay.
On the architectural level, however, we have that, because on the architectural level is when you put these things together, you decide about how things are interconnected.
Okay, well then so that would probably be the thing that we'd need is to see what is to see what the instructions are for how you change connections at the architectural level.
Okay.
If that's in a paper already, then that's a pointer. If that's documentation that is yet to be written but is in your mind, that would be very helpful.
Okay, so we will look into this.
Okay.
My understanding there is some of it, but it sounds to me that maybe it's not so visible. Okay.
Okay.
At least not so visible, not so obvious. Okay, so then we'll respond to this.
Okay, yeah, because then then there's it's not only just the C. elegans connectome parenthetically, but I mean like Porg also runs a resource called the open source brain where there's hundreds of models that could in theory be plugged in. So so implementing NeuroML is certainly one thing, and I think a good goal. But even prior to that, we might be able to get something really quickly just by better understanding how your existing connection system could be modified. So yeah, if that's a concrete thing that comes out of this, then awesome. I think that would be cool.
Very cool.
Yeah, no, this is a cool suggestion. We'll follow up on this.
Okay. Right
at least I'd like to understand the implications on this.
Yeah, yeah. I mean, that's the idea. Okay, well, so we did set this for an hour, and I actually have to run. So are we okay? Thank you very much, Aurel, for taking the time to do this. This will be posted up on YouTube as well. Folks, we can refer to it as we go. But let's keep the conversation going, and let's keep interacting. You guys have a mailing list, right?
Yeah, we have a mailing list.
Okay, so we should also sync up those lists. I'll try to send a note out to both lists then after this, just kind of pointing to this and make things happen. So
yeah, really interesting work. So thank you so much for talking to us about it today.
Thank you, guys.
Thank
Pleasure
you.
chatting with you.
Cheers.
Okay.
Talk later.
Thanks. Bye-bye.